Thank you. All right. So before I start out, I just wanted to do a thank you, especially to make sure that I don't miss that, have or run out of time at the end. Ann Huber from Soil Resource Group and Jen Llewellyn was with OMAFRA, is now a private consultant. They both supported this project in a huge way. COVID wasn't the only issue that created hiccups for this project over the last few years. I was part-time for quite a while. So they made this project happen. So I just wanted to make sure that I acknowledge them. So getting into what we found over the last few years, we established early on through the literature search and evidence that, you know, light and nutrient levels have major impacts on ponds and the water quality, on surface water quality. They're key drivers, particularly phosphorus and nitrogen. And there's issues, of course, for not only for the farm in terms of the quality of water for irrigating crops, but also the physics of it. You can have clogging lines, etc. And there's also regulatory and other pressures, environmental pressures, to make sure that your water quality is adequate. So, sorry, my screen is just giving me, I'm going to try to move some things out of the way so I can see. So we had a general design of looking at in-pond and pre-pond treatments, whether technologies or treatments. I'm using that term interchangeably. We had mostly cells, which I call mesocosms, within ponds where we would apply the treatments, but we did have some larger portions of ponds and full-scale pond treatments that were performed. We had a total, by the end of the project, we kind of kept accumulating sites. So we had 4 nursery ponds, 3 greenhouse floriculture ponds, and a couple of silos thrown in there for good measure. We did benchmark monitoring and preliminary work in 2019 to set up the mesocosms and how the trials would be set up. And then we did seasonal monitoring and analysis of those different technologies between 2020 and 21. And last year, technically, there was not a huge budget available last season, but we tried to squeak in a few things extra that to sort of establish what we thought was happening and where we thought the most effect would be. So we did water sampling of either grab or whole column samples for nutrient analysis mainly. We had an in situ multi-parameter meter that did oxygen, chlorophyll A and phycocyanin levels, among other things, and did some mind-boggling work, tons of microscope diagnostics of water samples looking at phytoplankton populations. And we did some limited sediment nutrient analyses as well. That wasn't as often as what we would have liked, but they were challenging to get samples. So we limited that analysis part. So primarily it was based on water samples, in situ, multi-parameter readings, and the phytoplankton diagnostics. And now it's giving me a hard time about going to the next slide. There we go. Mesocosms, I think I've shown some of these slides before. Just to give you a reminder, this is the column that we put into the water. They typically range from 2 to 4 meters cubed, depending on the depth of the water. We had a standard 5-foot diameter. I got really good at building these. So we had a variety of treatments that we tried. It's very difficult to do replicate treatments in small ponds. There's some really big projects out in Germany that have really massive mesocosms in acres and acre sized ponds, and we just don't have that kind of space. So we tried to do replicates where we could. We did aeration, some shade with knitted shade cloth in a couple of different ponds, Site B and Site C. And then we tried to do some replicate mesocosm work in 2021, where we had some bacterial populations for sediment degradation, both a couple of different types. One was called C for some reason, one's called pond. We did some combinations. And the wollastonate didn't really do a lot in terms of phosphorus binding, so we tried a different one in 2021. And so we did have a couple of replicates there. And in the site C, we basically repeated what we had done in the first year, because we'd had some hope that that would look good. So just a visual of what this would look like. At site B, there were, I think, 10 total mesocosms in the main pond. Each one we had a manifold in the center, that white object that you can see in the center, putting aeration out to all the different mesocosms. Just to keep it's important when you're applying bacteria and some of these coagulant type products to keep the water circulating. That's the partition that's in the same pond. It was about 15 meters cubed of volume, so a little bit larger. We thought maybe if we added a lot of bacteria, had aeration in there, and we could potentially have some more effect if we had a bigger volume, because we were concerned from the 2020 data that the mesocosms were not large enough to show a result. And the site C, their H pond, they had pretty standard treatments over the 2 years, although the shade one that's on the upper right here, the kind of gray, we switched it to a full solid landscape cover cloth in the second season of sampling. So we also did in pond, as mentioned. So I think from previous reports, I've shown you some phytolinx work that we did. We had mixed it up with some biocord, so for periphyton to develop on in 2020, and had quite a few challenges there. And so we did do some more, we did another set of sampling in 2021 on that. And we did whole pond work as well. At site E, a greenhouse floriculture farm that had aeration and nanobubbler system set up. And then they mixed it up quite a bit, added some water hyacinth plantings, which essentially covered most of the pond in that 1 year. And they also incorporated sonification. So it's a bit of a mess in terms of experimental design, but some interesting things being tried. So we wanted to track what was happening there. So just some visuals here. Upper left here is the base phytolinx frame. And in the center, you can see that frame, Anne's holding it up there, and you can see the planting well developed on top. And when it was installed on the bottom here, we had 3 separate channels, a center one with nothing in it. And initially we had one with phytolinx and one with phytolinx and biocord. So like a mesh system that improved periphyton development. And site E, that's the 3 treatments, the whole pond. So they had a nanobubbler, the water hyacinths, and sonification, a bunch of things happening there. And last summer, as I said, we had a limited budget, but we wanted to try a couple of new things. And we were 2 farms had said to us, we're putting in, we have full cover. One was just about to install it. The other one had a cover on their silos. They said, come ahead, go ahead and sample if you like. The one site never ended up getting their cover, their whole cover on their pond installed, which is unfortunate because we would have loved to see the difference. But we did get some interesting data from the use of bird balls. So that was pretty cool. And they're black balls, and that's the picture of the silo. Wide angle view, and you can see it's got a liner. There's a netting over top to prevent animals or birds from getting in. And then the bird balls, they're actually black, like 3-inch balls. They cover about 95% of the surface. And the pre-pond treatment, site A was something that we had worked on in the very first couple of years. We had some wood chip trials that we were doing to look at flow through and an attenuation of nutrients. Unfortunately, we had to move away from that site, but fortunately, a different farm invited us to come and put in a pre-pond hybrid treatment swale, which contained wood chips, slag, and gravel. So there's a view of it with the wood chips in the foreground, and you can see there's a bit of a line there, and there's a short section of slag, and then a gravel section to polish the water before. And this is all passive. There's no pumps or tubing here. It's just graded flow. So That's our experimental design in our sites. What did we find out? And I think I mentioned last year, we did struggle with the mesocosms. They weren't as effective as we'd hoped. Maybe they didn't have enough volume. Maybe the water quality is not representative of, in comparison to the pond itself and the controls. Maybe there was too much flow between and nothing really happened, or maybe there just wasn't enough time. There is a huge amount of data there, and not a lot to really bring to light. So I think that's why we really focused on the following for last season to look at some more of the bigger scale treatments. In pond with the phytolinks was disappointing. We had high hopes for this. The plants grew really well. There's been good results with higher nutrient waters, so very eutrophic ponds and even sewage ponds, things like that. But here we had really high levels. Initially, we had a nice layout, and you can see in the bottom here in 2020, the water level just dropped completely. They needed the water. It was a dry summer. We couldn't, there was, like you could see the biocord actually dangling in the air. So that really got no samples essentially that year. We tried again. We just put the phytolinks in one channel and had 2 control channels, and they were flooded out in 2021. So again, nothing, no real data. Great growth, not enough information here, and not enough standard, what we'd hoped for in terms of controls to actually make any analysis out of the data. So site E, again, another bigger in-pond setup. The whole pond had the nanobubbler. This is in the first year. We had a really interesting algal bloom that was visible. Blue-green, actually, sorry, mostly blue-green, cyanobacteria, not algal. So there was some concerns there. What could we do long-term to make improvements there? So they got very creative, and they had their nanobubbler going. But as you can see, in the second year, they put water hyacinths in, and by in about a month's time, it went from maybe 10-20% coverage to about 95% coverage. It was pretty intense. So I think I showed this last year, and I wanted to bring it back again. In the first year, there were a lot of this sort of teal turquoisey color. That's where anything was a cyanobacteria, so the blue-green algae, blue-greens, and then these yellow and brown are and gray are more algae types that are showing up. So quite a lot of the cyanobacterial blooms in the first year. And in the second year, when we had this huge covering with hyacinths, you can see down at the bottom that it really changed by September. The population, the phytoplankton population, really changed and moved away from the blue-green blooms, which was really, really fascinating for us, and kind of got our brains going. Interestingly, they tried regrowing the hyacinths in a greenhouse for maintaining them over winter, and then brought them back in late spring. I guess it was early summer. It was, I think, July by the time they actually got them in the water. They did not take off as well, and those blue-green blooms came back with a vengeance in last summer, which was, so it kind of was a reversal, right? We had an improvement, and then it reverted back. There's a lot of sediment in this pond. It holds a lot of nutrients, so there's it's going to be a long-term project to control and to manage water quality here. And just an example of some of the data that we got. There's a lot to share, so I kept it very light here. But it was interesting to look at, total chlorophyll A and phycocyanin levels. And we looked at the blue dots are 2020, 2021 are the orange squares, and last summer, 2022 are these gray triangles. And I think it's pretty interesting that when they had the best control in terms of the way the pond appeared, and they had the water hyacinth coverage fairly high, is when, generally speaking, it was a much tighter, lower level of evidence of phytoplankton and blue-greens. So then looking at the water silos and these ball covers, and we thought this would be a neat site to look at. It's not a pond, but it's still water source essentially for nursery and floriculture production. So there's a main pond that's the feed water, and then there it goes out to the outdoor crops, and then these two silos are return silos. And both silos have this bird ball and a net covering on it, but the pond is uncovered. So they're adding nutrients. Keep this in your mind. They are adding nutrients after the pond. So if we look at the phosphorus and nitrate levels, if we look at the pond, so we're looking at source water, so the blue line, phosphorus, total phosphorus, quite low. It's not perfect water, but source water typically in Ontario looks between, is usually around 0.3 to 0.5 and sometimes gets up towards 1 ppm. So it's pretty standard. That's pretty normal source water. And the nitrate is below detection limit, which is also expected. The return water, again, you would think they've added some nutrients. It's as water soluble fertilizer going out onto the crop. And this is coming back into the two silos. And you can see the two silos have significantly more phosphorus and significantly more nitrate, which totally makes sense, right? So what gets really, really cool is that the chlorophyll level and the phycocyanin levels are really low in those silos. And they're very similar to what you see in the pond. In fact, they're lower than what's in the pond. And that's, I think, pretty cool. That's a real indicator that covering has a major effect. And we never saw any sign of a bloom or any issues with the water quality. It always looked clear in the silos, and we know that there's nutrients there, so it was possible. And those bird balls have been in for, they were in 2021, which we didn't know, we didn't sample that year, and they were, and I guess they put them back in the spring in 2022, which is the year of this sampling. And I don't know how I'm doing for time, but I think I'm okay. Still have a few minutes, right? Jamie, you can let me know.
Yeah, you got a couple more minutes.
Okay, good. So we know it's back to like nutrients mainly. And so covering obviously seems like a really important thing, and nutrient supplies. But what would be the best thing would be to prevent nutrients from ever getting to the pond in the first place. So we worked with a farm to look at container runoff, container nursery pots that were top dressed, and on the left side at the corner there's an inlet pipe that comes from that area and it enters the pond. So we were able to compare the water level, nutrients coming into the pond and the pond water quality, and we compared it. It's pretty much in line with what we have seen in other container nurseries. This is an old project. We spent a couple of years monitoring nursery drain water, discharge waters, and knowing and recognizing this is total phosphorus levels, that they're typically a little higher, maybe between 1 and 2 parts per million, and pond water itself usually sits, as I said, below 1. So it was in line with that. The pond itself, total phosphorus 0.3, and there's a bit of a range there. Soluble reactive phosphorus, which of course is very friendly and helpful for phytoplankton growth, and a bit of nitrogen and so on. And when we look at the inputs, this is what's the discharge water coming from the container production. So that's where we decided to put it through the hybrid treatment system that Ann Huber's developed, where you combine a traditional wood chip bioreactor with slag and other components to achieve certain nutrient removals. And this is a shallow system. It's not going to work the same as a traditional hybrid treatment system. It's passive, but we were really hopeful that it would deal with some of those nutrients. So again, there's just a photo of it, and I'll quickly look at, I'm mostly interested in the results. So in 2021, we got some preliminary results that made sense. Nitrate was being removed. Some of the phosphorus is being removed. So phosphorus is the upper graph after the wood chip component, and obviously all of that nitrogen. So that was pretty exciting, the nitrate. Didn't look like cells 2 and 3, the slag and the gravel were doing a lot at this point. Sorry? I'm just getting some feedback. And in 2022, though, it actually, the nitrate is still being removed, and it does now look like we've got some phosphorus removal from the slag, which is what that was intended to do. So we were pretty excited about this. We are hoping to be able to develop this further, this concept, and work with some other farms to try this technology in the future. So the takeaway for a new pond, keep it clean. That last site had a new pond, and with basic drainage from container nurseries, we could see some change in the water quality, but the hybrid treatment swales are a potential solution. And for existing ponds, it's critical to prevent additional inputs of nitrogen and phosphorus when you can. And it really looked like covering a pond or even a silo can help limit that growth. Long-term degradation and organic matter and nutrient removal from sediment is a big project, but obviously it's going to be part of the picture. And product deliverables, we're on target for those. I won't put a lot of effort into reviewing that. And I want to say thank you again, particularly to COHA and their support through the project. As I said, it was not an easy time for me, and we still managed to get a lot of this work done. So thank you.
All right, thank you, Janine. Does anyone have any questions before we move on to the next presentation? Quiet group. I mean, the covering of the pond certainly makes sense, right? Without sun, chlorophyll doesn't operate. I'm sure there's more to it than just that, but certainly one of the easily implementable options for smaller ponds, I suppose.
Yes, and it's not, that's the trick. It's a smaller pond, so there's liner options, like you can do complete covers, and for silos and small ponds. For larger ponds, it's a bigger expense. So that's where we were hoping to look at phytolinks and some of the other options for covering. But yeah, and the water hyacinths are challenging. They can be invasive. So obviously I was using a shovel to actually get through the pond and be able to move the pedal boat through. So it's pretty, you have to be careful what you choose, but I think that it looks like that's the right direction. Keeping an eye on the sediments, preventing it from getting in as number one, keeping an eye on the sediments and what can you do there, and covering does seem to make a difference. We saw that in Ohio at another farm. They have a large pond that they invested in a plastic cover, and within months, about 6 months, they saw a huge improvement.